So let's see what this diagram is showing about. So it shows the evolution of the plant from over geological time scale. Here we can see the oldest plant, that is primitive, then modern plants that are advanced, and the time scale, that is from Silurian to Quaternary, and yellow line show the evolutionary pathway. So here we can see the origin of plants, which starts with the chlorophyte ancestor, that is green algae, meaning the plants originated in water, no roots, no stem, no vascular tissues. Then the first land plant then come like the tracheophyte ancestor, rhinia type plants, and Coelophyton. So what are the features of it? First, the land plants, their simple body start with the vascular tissue like xylem and phloem. Then there are earlier vascular plant diversification. From here, the different branches evolved like lycopods. These are the small vascular plants. Then Phenopsida, that is the horsetail, then fern, that is the pteridophytes, and important because these were the first plant with true vascular tissues. Still, no seeds are there and reproduced by the spores. Then the seed evolution. It is a major change where the progymnosperms will form the seed ferns here. The evolution of the seeds, that is the huge advantages here, the huge advantage and protection plus survival. So the gymnosperm, they are also known as the naked seed here. Example, the cycads, conifers, ginkgo, natalis, they feature seed not enclosed, that is a naked seed, and no flowers. Whereas angiosperm, they are the topmost advanced one. They are the final stage where the flowering plants, monocots and dicots are seen. Here the seed inside are fruits, flower present, and most successful plants today. So key evolution trend, you must remember this: water to land, then no vascular to vascular, spores to seed, naked seed to enclosed seed, and no flower to flowering plants. So here the one line summary for this is like algae, from algae to bryophyte, then pteridophyte, then gymnosperm, and then angiosperm. So at the bottom we can see the algae, then in middle we can see the fern and vascular plants, and at the top we can see the seed plants, and then finally the flowering plants. So here we can see a question: which feature of genetic material is considered the most useful specifically for the process of evolution? That is the slow change. Evolution rely on slow change, that is mutation in a genetic material to provide the raw variation that natural selection acts upon over the time. Next question is, according to the mutation theory proposed by Hugo de Vries, which one of the following state is not true? Mutations are random and directionless. Yes. Saltation single step, large mutation, true. Mutation are directional and gradual. This is wrong. Mutation is are random and directionless. It is given in option 1, that is true. Because of that we can see it is not directionless. Mutation cause speciation, yes, it is true. So correct statement for this is mutation, means mutation is not directionless and gradual. That's why we have to select which is not true. So here D worries possessed that mutation are random and directionless, whereas Darwin's variation are considered small and directionless. So how to learn the reptile evolution? Here we can see sleepy student take road, pick bread, drink Bonavita and meet the people. As for sauropsids, here we can see the sauropsids, then synapsids, that is SS, then thecodonts. Thecodonts, here we can see thecodonts, then theropsids, here we can see dinosaur, and birds. Here birds and mammals. Here in this diagram we can see the chemical constituent of protoplasm. Here we can see for plants and animals. So for plants there it is 75% of water. Fats and other are 1%, proteins are 2%, mineral solid are 2%, carbohydrate are 20%. Whereas if we see the animal, the protein, it is 15%, fat is 13%, mineral solid are 4%, carbohydrate and other are 10%, whereas the water is 67%. So you have to remember the percentage of the components present, like water 75 in plants, and in animals it is 67%, whereas the component of protein is 15% in animals and 2%, only 2% in plants, and mineral solid 2% and mineral solid 4%, carbohydrate 10% and 20%. That means the plant have more carbohydrate than animals, 20% in the protoplasm. This is the chemical constituent of the protoplasm. So we have to solve the match the pair, match the scientist in the image provided and their landmark contribution, select the correct option. So we know that here the Frederick Griffith, the experiment on rat, we know that the SR, that injections and all, so that is the transformation experiment. So we know that A pairs with the 5, so we have two options that pairs with the 5, that is B and D option. A is pairing with 5. Now gamma, here we can see double helix structure. Is it Watson or Crick or Meslin and Stale? Now it get confused now. Watson and Crick did the double helix structure or semi-conservative replication of DNA? Semi-conservative replication of DNA for Meslin and Stale. So here we know that D is pairing with the fourth option. So we get the correct option here, that is B1, because gamma, the sequence of the 3 nucleotide code for single amino acid. Watson and Crick, double helical structure. There is no anyone showing induced mutation. Yes, I am correct here. Griffith discovered the transformation. Gamma proposed a triple codon. Watson elucidated the double helix structure and Wilson proved the semi-conservative replication. So let's solve this MCQ. Here agarose gel electrophoresis result showing the result. Analysis of agarose gel electrophoresis result shows. Select the correct combination regarding the size of the DNA and fragments at the position A and B. Now you can see in the diagram here there are A and B position. There are whales, 4 whales present here. The A and B are present and they are your DNA bands are present in this. So we have to select the correct option here. So technical like recombinant DNA technology and agarose gel electrophoresis. Option A and B is showing the technique, and here we have to tell which one is large and which one is smaller. Now here we know that here the position, if we see the position of recombinant DNA technology, it shows larger, and B, it shows position B shows small fragment. Now how we can get this answer? Larger DNA fragment face more resistance in the agarose matrix and stays near the whale. We can see here, see the whale, and which one is staying near the whale. Whereas if we see the position small fragments, they move faster toward the farther end. Here we can see here it is moving toward the farther end.